Imagine a medicine that is based on prediction, where we can intervene at the time when still people are resilient. And so we're not going against nature, but we're going with nature. How more effective this could be.
Aging. Like gravity, it pulls on each of us. Why do some of us age without illness? How do our bodies and minds experience aging at the cellular and molecular level? What's the future of aging in our society? And maybe, most importantly, what can we do about it today? My name is Gordon Lithgow, and here at the Buck Institute in California, my colleagues and I are searching for and actually finding answers to these questions and many more. On this podcast, we discuss and discover the future of aging with some of the brightest scientific stars on the planet. We're not getting any younger, yet. Hi everyone, welcome to the show. This is a special treat today. I get a chance to talk to Luigi Ferrucci, and he's one of my scientific heroes for so many reasons that will emerge during the podcast today. And so I think this conversation is going to focus a lot on translation and what's going on with aging studies and interventions and what people can do. Luigi, again, thank you for doing this today. I feel like you're someone in demand right now because you're right at the heart of so much that people are talking about in terms of aging. But I do want to start just by going back to the fact that you're a geriatrician and an epidemiologist, and I'm kind of wondering how you got there.
I think that's a story, really. I was 16 years old, and I was serving dinner at the Rock Cross dinner, and I got a plate of spaghetti to a guy that was, on a table in a corner. And that guy looked at me and said, "It's Saturday evening. What the hell are you doing here? You should be out playing with your friends and going out with girls." And I say, "Well, I want to do something that is important. I want to contribute to society." And he told me, "If you really want to contribute to society, you have to study aging, because the people that will age in this world are already born, and they will change everything. Aging will change the society, the family, our economy, our healthcare. You have to get into that because people are not doing it." And so I decided to do that.
That's amazing. So this was someone who was needing help?
No, that was the person was somebody really important. The person was Francesco Maria Antonini. I discovered that later, which was the first chair of geriatrics in Europe. And so I decided early that studying aging was important. And then I was involved in care for a very, very, very long time. But as many of the geriatricians can tell you, at that time, geriatric care was not very famous. Nobody knew what the geriatrician was.
Yes.
And we had very little to offer to our patients. And so I decided that if I wanted to make a difference, I needed to do research. And, the rest is history.
That's an amazing story. So you went to medical school. I guess the other way would have been to do basic biological research, but maybe there wasn't so much around back then on biology of aging.
No, there was very little. I think that I had during my study of medicine a passion for the biology, and that was because my professor of basic biology was an incredible woman that really infused in me this passion. But then, I wanted to do something applicable, and so I wanted to see patients, and so that's why I went to medicine and then to geriatric. And our geriatric specialty was a little bit, peculiar, because Professor Antonini thought that in order to study aging, you need to understand human physiology. And the best way of understanding human physiology is to do acute care. So our internship was not in a nursing home, was in an acute care unit. I stayed several years in an acute care unit. And that was, my training is really in acute care. And I think it was important because you create these automatic responses, when you have acute patient, and that has remained in me and has been very, very useful.
And I guess you developed a comfort level with dealing one-to-one with patients as well.
It gives you confidence to talk to patients. It gives you confidence that if something acute happens, you know how to deal with that. And so I think that has been very important in my life.
So you moved into epidemiology, and was that a certain level of frustration that we simply didn't know the factors that were driving what you saw in the clinic?
So I wanted to do science, and at some point I realized that the weapon I have in my hand to fight the disease of aging were not enough. So I had an opportunity. There was an opening for a course of epidemiology of aging at the London School of Aging and Tropical Medicine. And so I spent a month there, and I spent a month there with the best of the best epidemiologists. I mean, Richard Peto was one of the people that were teaching, and Dr. Rose was one of the epidemiology. And because I had very little money, I used to go for dinner in this Indian restaurant, and they were passionate about Indian food as well. So we spent evening eating Indian food and talking about aging and epidemiology. And that developed completely my passion. And I came out from that experience, wanted to be in epidemiology, understanding public health. And that when I started doing some studies in Italy, I created a study that was called the InCHIANTI study that is still producing incredibly good science. And so I started doing epidemiology. Later on, I became the director of the Baltimore Longitudinal Study of Aging, which is really an amazing study. Progressively, I became more interested, in the dark side. I started wanting to understand mechanisms. When you've done a lot of observational studies, you start thinking, why this is happening? Why people, experience decline in muscle strength? Why people lose their memory? And in order to do that, you need to go deeper.
I'm not going to forget that you called mechanism the dark side. That's great. Well, let's talk about the Baltimore Longitudinal Studies. So this is very famous, and you're synonymous with it, and director and so on. Just for those listeners that are coming to this maybe new, describe the origins a bit about this study and the especially the structure.
I think that this was an intuition of the titanic force in the field of aging, which was Nathan Shock. Up to that point, people have done study of aging by correlating, by comparing young and old people. For example, is liver function higher or lower in old compared to younger people?
Yeah.
And there is nothing wrong with that. We still do a lot of cross-sectional studies, but there is a problem that people that are old also have experienced different things in their life. Somebody that is 100 years old now was born, during the war. And so imagine, what kind of nutritional status of stress they went through. So they're different from younger people, not only because they're older, but because their experience in life has been so different. The only way to really study aging was to follow people over time, and study them multiple times. And so that you can study aging at the single individual level, and so you don't have that bias. So the Baltimore Longitudinal Study of Aging is really the first study of aging that was done with this design. In 1958, the Baltimore Longitudinal Study started. So people were enrolled in the study, and they were enrolled in the study when they were extremely healthy. They had to be healthy because Nathan Schock wanted to study the difference between aging and disease. He said that one of the ways we can better cure for disease is to extract from disease the effect of aging. And we know now that was a wrong question, because now we know that there is no really solution continuity between aging and disease. But we had to discover that when our technology improved over time, we discovered that it was almost impossible to dissociate aging from disease, from chronic disease. But at that time, that was the construct and the idea. So in the Baltimore Longitudinal Study of Aging, people are coming to our unit every 4 years if they are less than 60 years old, every 2 years between the age of 60 and 80, and every year after the age of 80. And we host them. We host them for 3 days, and we measure the measurable. I think that any measure that comes to your mind, you're probably doing, including a dedicated MRI that we have in our unit only for the BLSA. 2 actually. Now we'll be using a 3T MRI, and now we'll be using an even stronger MRI, 7T MRI. And that is a really new emerging technology. And I think that allow us to really understand a lot better, not only how people age, but how the different dimensions, are correlated to each other. And we found that the people that lose muscle strength faster are also those that tend to lose the kidney function faster or heart function. So there is some sort of homogeneity in the way we age. And that really convinced me of the Geroscience hypothesis, that there is an underlying force that is driving these changes that we can call aging or biological aging or whatever we call it, is there.
Thank you. There was a couple of interesting points you made along the line there. So first of all, Nathan Schock, I guess, was the founding director of the National Institutes of Aging, which is kind of an important development, and the federal government's commitment to all of this, along with the Baltimore study itself. And then you mentioned the fact that Schock was interested in this, if you want, separation of disease and aging. And I mean, I guess this was important at the time, right?
Yeah, absolutely. I think that Nathan Schock was certainly a visionary. It was, conceptualized the idea of longitudinal study before anybody else. But remember that at that time, medicine was organized by organs. You study myocardial infarction, you study, cirrhosis, you study hip fracture, and those were supposed to be disease. And disease are more likely to occur with aging, but they're not aging because some people do not develop them. Some people get to age 100 without having a hip fracture. So people thought that they were not connected. Well, the problem is that when you start doing longitudinal studies, you find that the same atherosclerotic severity that eventually lead to myocardial infarction occur with aging in everybody, almost in everybody. Mm-hmm. The same osteoporotic process that eventually lead to hip fracture almost develop in almost everybody. So the cleavage between the disease and the effect of aging is become very artificial, especially now that we have the technology to look at it. And then by looking at longitudinal trajectory of these functional organs, we realize that aging is pervasive across different organs. And the fact that aging is manifested differently in different individuals, it's probably due to genetic predisposition and also to the fact that we have different environmental exposure. Somebody exercise, somebody doesn't. And so the same aging process, will have a different effect in somebody who exercise and somebody who does not exercise. And this is so true that now that we are playing with biomarkers, we see that the signature of aging, it's universal. We find that some of the protein that change with aging in the blood change in the same direction for everybody. And when this change more than in others, they are predictive of the development of disease. So we read indirectly the biology of aging through biomarkers. So that's where we are at the verge of transforming the science into clinical application.
Yeah, that's super important. Actually, I want to talk a little bit more about technologies because, as you said, when your subjects come in for study, you measure everything you can, and everything you can is an ever increasing universe of potential techniques. And I'm wondering, just thinking about, I guess genetics was a major factor for a long time in terms of association studies and trying to find genes with main effects. And then we had the omics have kicked in a big way. We're talking a lot about clocks, which is related to biomarkers, of course. But let's go back to the genetics for a minute, because I think this is fascinating, because we've talked a lot about the effect of genes on aging in this series, and especially in model organisms, the effects can be absolutely massive. And then we come to the human populations, and things are a bit more complex. And I guess this could be summarized by the missing heritability question. Talk a little bit about how you study the effects of genetics on life expectancy and on individual diseases, and whether you think that we've that sort of well is dry now, or there's still things to be discovered.
You're making a really strong point of the missing heritability, where we all started doing genetics, making the assumption that study genetics will solve everything. We will find what are the genes associated with different trajectories of aging, and then we will find, based on genetic characteristic, what are the interventions that are perfect for a certain individual instead of another. That was the concept, and that was real into it. I do remember, doing the first GWAS that we did in BLSA, where we found that there was a peak on ApoE4 that was the ApoE4. ApoE is the strongest, the variant of ApoE, the strongest risk factor for Alzheimer's disease. And we found that even in BLSA, in the preliminary study, that in this figure, where each gene is represented by some sort of a tower, what we call the Manhattan plot, the ApoE4 was on top of everything else. And so that gave us a lot of hope that will happen. At the beginning, happened what has been happening always inside. So people kept their results separated, that they wanted to, do their, GWAS. And then they realized that nobody had enough data to say anything. And so the only way to really explore genetics was to create consortia, to have a big group that, regroup many different institutions so that the number were sufficient. Even when we did that, the association studies were many. There were many traits associated with genetic variation, but the amount of variance explained, the amount of association explained was minimal. And that is a problem, because when we study twins, monogenetic and polygenetic, monozygotic and heterozygotic twins, we find that some traits are very genetically connected, 30%, 40%. In GWAS, you're lucky if you find the 1% of the variance is explained by genetics. So there's this discrepancy between the two that we still, I don't think we understand completely. I mean, I think epigenetic is part of it, and it's becoming more and more important, but still, I don't think we have the key to understand that.
So going back
Yeah.
to GWAS, so GWAS stands for Genome-Wide Association Studies.
Yeah, in order to do that, you collect blood, you extract the DNA, the nucleic acid from the blood,
Yeah.
you come to white blood cells, and then you use a chip that look at genetic variation at different part of the DNA. In the beginning, we were using chips, so we were measuring 450,000 location. And then we got to 1 million to 20 million. Currently, we're doing what we call whole genome sequencing that allow us to really study the entire DNA in high detail. And then when you have that, to say, okay, who of these people is developing myocardial infarction? The genetic does not change. And so you see what is the difference of the DNA of people that develop myocardial infarction and those who do not.
Well, the big picture here, I guess, is that you said that the effect of genes is less than you might have expected. You mentioned 1% at one point. That's unbelievable. Just to sort of translate that for a particular condition, only 1% can be assigned to differences between genes between people. And then so for life expectancy itself, so the big picture, I know you're not the only person doing this, but for life expectancy at birth, which I guess is the big picture of what's the effect of genes on aging. What do you think heritability is at, the effects of genes?
The interesting thing is that the gene associated with extreme longevity are not many.
Yeah.
And the most constantly found in all the studies, ApoE2, which is the protective variant against dementia, is also the gene most associated with longevity. And then FOXO3A is the other one that everybody is finding. There are many other that I found in certain population, but not in other, but much more not very constant and not very replicable across different populations.
Yeah, no, it seems surprising considering the genetic effects on flies and worms and so that are massive. Although it's a different question in a way. You're asking about the differences between individuals, and those other big genetic studies are really talking about effects on populations that have variants at a single locus or a single gene. So I guess it's maybe a bit apples and oranges. But doesn't this all come back to what you mentioned originally about life experience, and really an appreciation that is the overwhelming force that's driving our aging as individuals?
Yeah, I think that why we all understand genetic, because genetics where all the information to build our body and make it function is there, so that why the DNA does not change over our lifetime, and so we're fixed machinery, we can modulate the way this machinery works. And the modulation occur through mechanisms that we call epigenetic. We don't change the DNA sequence, but we chemically modify the DNA in a way that respond through gene expression different to different stimuli. For example, this is especially being studied in the early part of life, where if you have, if you don't have enough nutrients, you change your gene expression to slow down your metabolism. So you need less nutrient to maintain life. And then being strongly hypothesized, and there is some strong evidence that these occurring in early life is a determinant of some of the metabolic disease that we see in late life. But then in late life, you get in a period where you can actually have enough nutrition, but your body has slowed down the use of those nutrients. And so those nutrients accumulate without being utilized. And that is one of the reasons why people develop diabetes or metabolic syndrome or other metabolic disease. So the experience, the exposure to environment, your behavior, the smoking, the type of nutrition change this epigenetic modification and so make us completely different in reality.
I've been trying to think of a metaphor for this. It's a bit like moving into a new house and there's a garden there. And the garden has a certain structure that it always will be. There'll always be a hill there, or probably there's going to be a path there. But you can decorate that garden and change it in ways where it looks completely different, despite the structure being the same.
Rafael De Cabo, did a study in collaboration with the BLSA, and he said, well, let's take a mouse that is genetically identical. I take mice that are genetically identical, and then in midlife, I look at the variability of something like, weight, or height, or walking speed. And what he found is that the variability of these traits is similar to the variability of human in midlife. Mm-hmm. So that shows you that genetic is a primer, but then on top of the genetic, epigenetic modification make us, each one of us a different individual. So you don't, even when you have the genetic identity, you don't have identity in aging.
This is an incredibly important result when we think about interventions, which I want to come to very soon. The natural variability between individuals that have the same genes is enormous. It's amazing, and something I guess we don't quite understand. I mean, all these animals are living in the same place, right? It's not like a human population at all.
Same diet, same everything.
So we're at a point where we've got all these technologies, and just to summarize, what do you think is the best way to measure aging right now?
I think that because we started with methylation, I think that that's the best way to study aging at this point. Yeah.
So this is a modification of the DNA.
By adding a methyl group, traditionally we think that when you add this methyl group, you kind of block, gene expression. That because you interfere with the binding of a transcription factor, and so that modulate, your gene expression. But we know that things are a lot more complex than that. It's probably true in different small parts of DNA, but it's not a general mechanism. But the bottom line is that Steve Horvath was the first, maybe not the first, but the first, well known, that discovered that if you study the methylation of a limited number of these binding sites in the DNA, you can create a score, 353. You can create a score that is associated with chronological age. I want to stop for a moment here, and I say something that I always say. If you find a drop of blood on the floor in your corridor, and you study methylation in that drop of blood, you can say the age of a person that lost that drop of blood with the precision of 3 years, 1.5 plus or minus. This was not possible before. We didn't have any biological marker that will allow us to know from a drop of blood age of that person. And it's an extraordinary discovery because since then, we only were chronological age. And we knew that people in their 70s, some of them are in bed and they can't really walk. Some of them still run, half marathon like me. I'm almost 70 and I still every year run half a marathon. So chronological age doesn't tell you, very much. Well, tell you something, but not enough about the health of an individual. And we hope that by studying this aging clock, methylation is one of them, we can understand more about biological aging. And I think that since the first methylation clock from Steve, others were created, some of them based on longitudinal data, such as the pace of aging from Dambersky and Grimage from AKEA, and they seem to be more powerful. Again, they're not powerful enough to be applied to clinical practice. And mostly because this has been applied in single population. We need to study them in many, many different populations. And then the other problem with this is that why we know they predict outcomes such as mortality, we don't know why. We have absolutely no idea of what is the underlying mechanism of the epigenetic clock. So the new clocks that we're now creating and people are creating that are based on proteins are probably much more likely to be applicable in the clinic because we know what proteins do. And I'll give you an example. I think that, I became a physician in 1980. And in 1980, we were measuring the same 32 protein when everybody was any patient was coming to the hospital. You study, the list, albumin, globulin, and then, the same markers. And we're still studying them, but our technology allow us to study 10,000 protein in less than a cc of blood, 10,000 in a very, very small amount of blood. And we need to leverage on that clinically because there is so much information in there. And in order to extract that information, we will need to do this measure in many large studies across different diverse type of population. And that is the medicine of the future. I mean, if I want to make a dream, I think that any patient coming to the hospital, we will measure them and then say, yes, you're coming here because you have a shoulder that is painful. I can see from your biomarkers that you are having occlusion of the left artery of the femur. And you need to do something about that now, because now our treatment will be effective. In the future, it will be a lot more difficult to do it. Imagine a medicine that is based on prediction, where we can intervene at the time when still people are resilient. And so we're not going against nature, but we're going with nature, helping the normal mechanisms of health in nature, how more effective this could be.
So I guess another measure is functional outcomes, things like frailty measures. What's your thoughts on those?
My conceptualization of aging, as you know, is the ratio between damage accumulation and resilience strategies. So the stochastic force that create damage accumulation in molecule and in cell, and then there are evolutionary selected resilience strategies that prevent or repair this damage. To me, frailty is when the effort of life is too much for the residual resilience that has remained. Even living is too much of a stress because you have exhausted your resilience strategy. So everything that you do determines accumulation of damage very rapidly. And that's why people, when become frailty, have a very, very short survival. Now, walking speed is another important measure. And to me, we know that it's incredibly powerful in terms of, prediction of mortality. It's more powerful than any other medical measure we know about.
Yes.
But I'm not surprised by that because, walking and bipedal walking has been the characteristic that allowed humans to survive in spite of, the aggression of other species. You are bipedal and then you free up your hands that allow you to do other things. So because of that, walking is incredibly physiologically redundant. You have many, many compensatory strategies that may allow you to keep mobility in spite of a lot of damage. And so because of that, when you lose mobility, when your walking speed is going down, that means that all those strategies are gone, and we can spend another hour, to explain what those strategies are, because it is one of the wonders of physiology of life. But imagine that when you lose that, that means that all the redundancy that was in your body is no longer there. So that's why it's so much predictive of mortality.
Do you believe in blue zones?
I think so. I think I don't know whether the blue zone are the one that have been identified. I was involved. I was involved in one of them. When I was a young geriatrician, I was an interviewer in Sardinia, where we identify. In fact, I was part of the first paper describing it. The area in Ogliastra, close to Nuoro, where there was a concentration of centenarian, and especially male centenarian, that is really very, very peculiar characteristic. I think that you live in a certain area where you have certain constraint of exposure, and those concentration of exposure and behavior can enhance your capacity to live longer and healthier. So I think that is I don't exclude that there are blue zone, and probably there are much more many than those that have been identified. But if that is true, that means that we should be able to develop drugs that do the same thing.
Yeah.
That's our hope. And
Well,
is rapamycin, is metformin, perhaps. I don't want to sell, these certainties, because they work in animal model. We don't have the result of the trials. The senolytics, all these drugs, seem to have a good effect in model organism, and then now many of them are in randomized controlled trials that will give us the unbiased answer about we can use them, to really enhance our ability to age well. And I hope they will work. But they may not work at the beginning. We need to be aware that some of this trial will fail, and then we'll keep trying. We'll refine them, and eventually, I'm sure that we will find them.
It's nice to hear that level of optimism from someone who is such a careful and conservative commentator on the field. What is the standard that we need to establish that a nutritional intervention, say, or a drug-like molecule is really slowing the measures of aging? I mean, do we need new populations? Do we need new study designs? I know people are talking about, Mendelian inheritance.
That is a great, question. I will respond in two ways. One is, hopefully the biomarker of aging will become so strong and predictive that we can consider them as proxy outcome. In order to study an effect of a drug on longevity, if you are study somebody that is 40 years of age, you will have to wait 45 to 50 years. And that's not practical. But if we are confident that those biomarkers of aging truly follow, the pace of aging, then we can see whether in a couple of years or 3, you're slowing down the pace of aging. And you can imagine that those proxy measures will predict longer survival. So I think that our ability to test what we call the gerotherapeutics is strongly binding to our ability to develop strong biomarkers.
Okay.
I think that is absolutely true. So I talk about randomized controlled trials because they are the gold standard. So what is a randomized controlled trial? We come very conceptually. Is to say, I take a group of homogeneous individuals, people that are between the age of 40 to 60, and and then I say, I select half of them by chance, by pure chance. I become blind. I put their name in a hat, and then I put my hand in the hat, and I put them in two groups just by taking that from the hat. And then
Yes.
I say, I ask those in group A to take a certain drug, and to group B, or a certain intervention, drug, whatever intervention I'm talking about. And
Yeah.
group B, I give them a placebo. A placebo could be, the same pill of color and shape, but doesn't have the compound that I've given to group A. Why this is important? This is important because health is a lot in our mind. So if you feel that you have been treated or not treated, that is going to influence your health. And
Yes.
so everybody needs to feel the same way. Everybody should have the feeling that they have been treated with something that is good, although half of them don't know, but they're taking a pill of sugar. And then, then you follow them for a certain outcome and see whether those who have taken the intervention or the drug do better than the other. In this way, you have an unbiased response that tell you that something is working or not working.
First of all, it must be difficult to think about a placebo for your Southern Italian cooking, but maybe you can work on that. But the placebo effect is absolutely massive, right? I mean, it can be 40% of the benefits you see in most studies. So, and that's something that I think we need to be very cautious about. There are study designs alternatives like the N of 1 type study. Do you have any feelings about this? This is where really you're studying individuals and you're making changes and you're following those changes, I guess, is the way to summarize it.
Over time, yes. I think that they're becoming very popular under the idea that each individual is different from the other. So the only really way to understand the effect of intervention is to follow individual over time. I think that why I like them. I think that the regulatory agencies still don't recognize them as rigid enough. And so I think that it's important that we stick in this stage, with the traditional approach. But I think that for research purposes, certainly the N of 1 are very, very interesting, design that need to be explored.
What are you really excited about right now?
I'm excited about the study of aging. Why? Because I think that we have made a massive effort to understand the mechanism of aging. But if you think about it, what we have found are not the mechanism of aging. The so-called hallmarks of aging are all mechanisms that oppose aging. Mitophagy, mitochondrial function. And so what we have discovered are the mechanisms of life. Those are the mechanisms of life. And only enhancing the mechanism of life, we can enhance the health of an individual. So I call them the mechanism of intrinsic prevention. We have fantastic mechanism of intrinsic prevention within us. And if we could enhance them, that will be much more powerful than any extrinsic prevention that we have done so far. To me, that is the future of medicine, because I think that the prevention that we have done so far is kind of hitting a ceiling. We're not seeing in the population very much more improvement. And so I think that changing the direction and acting on enhancing the mechanism of intrinsic prevention, it's very hopeful for a better future where we compress morbidity. I don't think that extreme longevity is something that I care very much for, but compression of morbidity where everybody live their life fully and then in a short period of time they get sick and die. I think that's the ideal situation that we want to accomplish. And I think we can. There is some evidence that we can.
Thank you. That's a really thoughtful and thought-provoking summary.
Luigi, it's delightful to talk to you. I could talk to you all day, but maybe we can share a meal sometime soon.
Absolutely. It would be great.
Thank you for listening. Please subscribe, share, and give us a 5-star review on Apple, Spotify, or wherever you get your podcasts. We're Not Getting Any Younger Yet is produced by Vital Mind Media. The Buck Institute's very own Robin Snyder is the executive producer. Wellington Bowler is right next to me here, directing the recordings. Stella B is behind the scenes, ready to debrief when we wrap. And the esteemed Sharif Izzat weaves the show together for you. If you're listening to the podcast, you'll know that there has never been a more exciting time in the research on aging. Discoveries in the lab are moving to the clinic to help us all live better, longer. The Buck Institute depends on the support of people like you to carry on our breakthrough research. Please visit us at buckinstitute.org to learn more and to donate.